Well, good evening, everybody. Gary actually called me about 30 minutes ago and was like, we got to do some extra songs tonight. I'm going to be doing Pew Packers. And he's like, you got the lesson, right? And I was like, that's right, I do have the lesson tonight. I've just been working continuously and forgot. But I've got something lined up for us I think you guys are going to really enjoy here tonight. It is going to be a little bit of a deviation from the way I generally like to teach. I usually like to teach very Bible intensive, using lots of scripture. Tonight's going to be the opposite. We're going to study something around apologetics. Caleb, do you have any idea what apologetics means? Can anybody help him? What does apologetics mean? What do you have? Some kind of a cheat sheet there, or what do you got?
She has a lot of notes.
Oh. Apologetics. Who knows what apologetics is?
Reasoned arguments.
What'd you say?
Reasoned arguments.
Reasoned arguments, that's right. So apologetics in regards to religion or Christianity are evidences or reasonings that we can make about Christianity. Why do we believe what we believe? What's our reasoning? What are the evidences for it? And I'm going to show you something tonight that's really interesting. And it's, most of you know that my degree is in biology. And one of the classes, the last classes I had to take at ASU was evolutionary biology. And I remember going into that class thinking, I am going into the lion's den. I do not want to be in this class, because I had already had some really negative experiences with professors at ASU in regards to Christianity. I remember it was my genetics teacher, huge 100 level class, my first year at ASU, hundreds of kids in here. And the first thing he said to everybody is, if you're one of these people that believes in religion, you need to stand up and you need to leave this class permanently. This class is not for you. You might want to think about switching your major. And I remember thinking, okay, wow, what a jerk. And I had to sit there and just, bite my tongue that entire semester at ASU in his class. That really aggravated me. Then my very last year, one of my very last classes was evolutionary biology. And the whole point of the class was to try and interest undergrad students into continuing on in master's and PhD programs in that field. And this was about evolutionary biology. And what they were trying to do is show students at the undergrad level what are some of the areas where we need help with research. And in this case, it was with evolutionary biology. And essentially the entire class, it ended up being a fantastic class for me, because the whole class was about, these are the gaps in the evolutionary biology theory, the Darwin theory about evolution. And it was just every class, we'd study and read all this material and go in, and it was a very small class. It was only about 8 of us because it was a 4th year class. And we would discuss it, and the teacher was very helpful. But I left that class thinking, how can anybody buy into evolutionary biology? There are so many problems with this theory. And one of the ones is going to be something we're going to talk about tonight, which is the idea of irreducible complexity, it's called. Irreducible complexity. I'm going to show you a video. It's going to be about 14 minutes long, but I want to give you just a little tutorial about what we're talking about. When I was in high school, it was so cool when we were in biology class, because they gave us a microscope and they gave us slides, and we got to make up our own slides, and we put little drops of liquid on the slide that had bacteria in it, and then you put a little slide cover on it, and you put it underneath your light microscope, and it had you could turn the dial on it. You could go from 5x to 10x, and then, man, if you could see it under 30x lens, you're like, oh wow, this is really cool. I can see what's going on in there. But what's one of the simplest building blocks of human biology when you're looking under a microscope is a cell, right? A human cell, or a bacteria, right? And so when we would look at it under the light microscope, and even today, light microscopes, by the way, have about a maximum of a 400x magnification, and that's with the brand new ones. And then if you use certain oil immersions. You can increase it maybe to 1,000 magnification with a light microscope. You're flooding whatever it is you're looking at with light. The light is penetrating it. Some of it's reflecting back at you. You're able to see what's reflecting back. But the problem is that it's very hard to distinguish when this structure inside what you're looking at ends and when the next one begins. They call it the resolution is not very good. Then, starting in the 1930s, they came up with electron microscopy, which instead of blasting what you're looking at with light, you're blasting it with a stream of electrons, which are much smaller, really, than light. So, guess what? Anybody have any idea? Now, that technology has improved until about the year 2000. I'm sure they're still working on improvements. But as of the year 2000, anybody have any idea what the maximum magnification is of, they call it transmission electroscopy?
10,000.
10,000? Anybody? Any other guesses?
1 million.
1 million?
100,000.
100,000? 50 million. 50 million. The detail that we can now see inside the human cell or inside a bacteria has vastly changed in the last couple of decades even. It is on a scale unlike you can even imagine the detail that you can see. And what we have learned is that there is so much more complexion to a simple one cell or a simple bacteria than we ever thought possible. And this presents a problem for the concept of evolutionary biology. For example, what is the main principle of evolutionary biology or Darwin's theory of evolution? What's kind of the basics of that idea? What did Darwin say? Anybody have any idea?
Cells just keep multiplying.
They keep multiplying, and sometimes what happens?
They change.
There's some random changes, and sometimes it's because of errors in the duplication of the genetic material, and you end up with something new. And if whatever happens to come out of that engine that's new has an advantage somehow, then that might allow that whatever that organism is to have an advantage for survival. And then it could reproduce potentially that same mistake, and now you got a whole bunch of these things that have some kind of an advantage, right? Now, what if you produce something that has no advantage? You make a mistake in the genetic copying of material, and now you have some new thing that's formed inside the cell, but there's no advantage to it. What happens?
Strongest shall survive.
The strongest cell shall survive. So if there's no advantage to a mistake, that cell is going to die just like any of the others. There's no advantage there. Eventually, that's going to be lost. When it comes to bacteria, I used to think it was so cool because we look at, in high school at the microscope at our little slides, we would see the bacteria, but then some bacteria were scooting around on the slide. They were still living cells that could move. How would they move? How do they move?
Flagellum.
A flagellum. What's a flagellum, kids? It's like a tail. It's like a tail. And you'd think, well, what a simple thing. I can imagine how a bacteria at some point in the distant past, a mistake happened and suddenly, bam, there's a flagellum, now a tail, and now what kind of advantage does that give it? Well, it can move around in whatever medium it's living in, and it can get to more nutrition, it can have a better chance at survival. And so when you think about it, that's like the most basic, simple thing that you could think of in biology. Well, one cell goes to one cell with a flagellum or a tail to help it scoot around and get to stuff. But when they started being able to see in detail under an electron microscope at 50 million magnification, it's incredible what we learned. We learned that the flagellum actually has a motor inside there that's running it. It has It has a U-bolt, that makes the angle right on. It's got little clips that hold it onto the outside of the cell. It has 40 different structures, actually, that have to fit together to make this thing run. And what happens if one of those 40 pieces that builds this motor isn't present? It doesn't work. It doesn't work. It's like what happens on your outboard motor on your fishing boat when you take the propeller off. No propeller. It doesn't go anywhere. It doesn't work. So what this tells you is that it's not just a tail appears there one day. You have to somehow come up with 40 different unique structures that build this motor that drives this cell someplace. And they would have all had to have been present, created, manufactured in the bacterial cell all at the same time and in the right order, and they would have to be fit together. And how would that happen? Now you can imagine, well, maybe one or two of them could have accidentally, through genetic copying mistakes, have been formed. But what about the other 38 of them? And how did they get fit together all at the same time? So even if you created a couple of the components, let's say that the cell accidentally created the propeller, but there's no engine. Well, so what? It doesn't give the cell an advantage, right? So that's what we're going to be looking at. This is called irreducible complexity, and he's going to compare it to a mousetrap, a simple mousetrap. Same thing. You got a spring, you got a little retention thing that holds the trap back, you got the thing that flips forward, you got the piece of wood it's mounted on. Mousetraps can work really good, but if you're missing one of those pieces on the mousetrap, it doesn't work. It doesn't work. Ask the wolves, they know. So let's check this out. I may have to run back there and enlarge this with the mouse, he said. Let's see.
For the longest time, I believed that Darwinian evolution explains what we saw in biology. Not because I saw how it could actually explain it, but because I was told that it did explain it. In schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution: A Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before. And the arguments seemed pretty convincing. And at that point, I started to get a bit angry because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and since have decided that Darwinian processes are not the whole explanation for life.
Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy. When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
In evolutionary terms, you have to be able to explain how you can build this system gradually when there's no function until you have all those parts in place.
But could Darwin's small, favorable variations have produced a bacterial flagellum? Some scientists doubt the possibility.
How could something new, like a bacterioflagellar motor and all of the components that go with it, how could it develop out of a population of bacteria that don't have that system? When each change, according to Darwin's theory, has to provide some kind of advantage.
Imagine such a scenario early in the Earth's history. An evolving bacterium somehow develops a tail and perhaps even the pieces necessary to attach it to the cell wall. Yet without a complete motor assembly, this innovation would provide no advantage to the cell. Instead, the tail would lie immobile and useless, invisible to natural selection, which by definition can only favor changes that aid survival.
The logic of natural selection is very demanding. Unless the flagellum mechanism is completely assembled and actually works, natural selection simply cannot preserve it. It cannot be passed on to the next generation.
The important thing to realize about natural selection is it selects only for a functional advantage. In most cases, natural selection actually eliminates things that have no function or that have a function that harms the organism. So if you had a bacterium with a tail that didn't function as a flagellum, chances are natural selection would eliminate it. The only way you can select for a flagellum is if you have a flagellum that works, and that means you have to have all the pieces of the motor in place to begin with. So natural selection can't get you the bacterial flagellum. It can only work after the flagellum is there and operating.
In 1996, Michael Behe published a book titled Darwin's Black Box. In it, he argued that natural selection, Darwin's designer substitute, could not explain the origin of the bacterial flagellum or any other irreducibly complex biological system. Instead, Behe concluded that the integrated complexity of these systems pointed to intelligent design. Darwin's black box created immediate controversy. Over 75 publications, including some of the world's leading newspapers and scientific journals, reviewed the book. Some scientists praised Behe's work, while others dismissed it as unscientific and religiously motivated. Behe's critics also insisted that he had underestimated the power of natural selection. They argued that the flagellar motor could have been constructed from parts used to build simpler molecular machines, like this needle-nose cellular pump. If the components of the pump already existed, they could have been preserved by natural selection even before the bacterial motor arose. This theory is called co-option.
It's essentially saying that evolution or natural selection at some point was able to borrow components of one molecular machine and build a new machine with some of these components.
Scott Minnick has studied the flagellar motor for nearly 20 years. His research has led him to challenge the co-option argument.
With a bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually, you're going to have to account for the function of every single part as originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity. For its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now.
Okay, I think we get it. You know, when they first illustrated, and I first saw that illustration of the motor, that's just truly amazing. I mean, it looks like an outboard motor on a fishing boat. It's that complex. And so Darwin had no idea that that existed. And I wanted to read to you, you know, some of the most important scientific discoveries ever in the world have been made by Christians. And it's because they expected that there was order in God's creation. There would be patterns that they could look for, and they would anticipate, they would theorize that, well, if this is patterned like this, then we should see this at the other end. And I want to give you something that Albert Einstein said. This is Einstein saying this. We are in the position of a little child entering a huge library whose walls are covered to the ceiling with books in many different tongues. The child knows that someone must have written those books. It does not know who or how. It does not understand the languages in which they are written. The child notes a definite plan in the arrangement of the books, a mysterious order, which it does not comprehend but only dimly suspects. That, it seems to me, is the attitude of the human mind, even the greatest and most cultured toward God. We see a universe marvelously arranged, obeying certain laws, but we understand the laws only dimly. Our limited minds cannot grasp the mysterious force that sways the constellations. Let me just read you a few names of some of the people who have made some of the most important discoveries. You'll recognize them. These were all Christians. Gregor Mendel, Galileo Galilei, Michael Faraday, Pascal, Newton, Kepler, Leeuwenhoek, Kelvin, Dalton, Babbage, Boyle, Morse. All of these guys were Christians, and most of them assert that their inventions or their discoveries would have never come about if they did not have the knowledge that the world has a design. And there must have been a creator, a designer that designed it. And that was what led them to many of their discoveries. And so I find that to be extremely interesting. This is a very difficult argument to deal with, irreducible complexity. If you're ever dealing with somebody that is so stuck on evolution, this is a great video to show them, because it's very, very difficult coming from somebody with a biology degree, if they have any actual knowledge about biology, to argue with this. And what we're seeing is that biologists and people in the scientific world, as we grow a deeper and deeper understanding of the material world, it becomes harder and harder to deny the existence of a divine creator or designer, because it's so much more complex than we ever thought. Things seem so simple in the day of Darwin, and now we have a 50 million magnification instrument to look inside the cell, and we're like, wow, this is a lot more than just a tail that grew on this one cell. There's a lot more complexity here than we ever knew, and the idea that this could just accidentally have developed through, well, a tail just developed. Well, it's a lot more than a tail. It's a tail and 40 different things that put it together, and it's got to be ordered in the right order. Who wrote the order? Who wrote the order of assembly? Somebody had to write that code. God wrote the code. So this is a great video. You can just YouTube it. YouTube, Google it, go to YouTube and search irreducible complexity. And this video will come up, or a couple of different formats. There's a little bit shorter version of it. Any comments or thoughts? Jerry?
Just completely coincidental. I was reading an article from Apologetics today on our new app on not this concept, but just evolutionists, and why there's 4 different reasons that this article states that refute Darwin and refute evolution and everything.
Yeah.
And I'll have to reread it again to remember every specific thing. But the funny thing is, all the experts, all the scientists, all those that are renowned in that field will all say, we don't really know how this happened. We can't explain how this would change into this, because it didn't.
Right.
They all have the same answer. They don't really know, but they go with it.
Well, you know, one of the things that Tyler, my oldest boy, he would confound his biology teachers whenever they would run through this stuff in biology class, was that most of the people that are teaching biology or evolution, I should say, don't even understand the difference between what we call microevolution and macroevolution. Does anybody understand the difference between that concept? That's really important to understand.
Changes within a population and changes outside of the population.
Right. So, for example, Darwin studied those finches, right? And he wrote all about these finches that he observed on the Galapagos Islands. And what he found was that different populations of these birds, these finches, had different shaped beaks, depending on what food source that they were going after. So some of them had a little bit longer beak, and that enabled them to get into this flower that they had to reach down a long ways in to get the pollen or whatever. And others had shorter beaks because the food supply didn't require them to have a longer beak. But regardless of the beak size, it's still a finch, right? It's still a bird. It didn't convert into a new species of animal. So that's called microevolution. Just like we can breed two dogs together and we can come up with a little bit different dog. Well, this dog's got a different color coat and he's got longer legs now, and it's still a dog. That's microevolution. No one's denying the concept of microevolution. It happens all the time. We see evidence of it all the time. My brother breeds Doberman Pinschers, and he's always trying to blend, this mom and this dad and create a better Doberman Pinscher dog. It's got a little bit better looking head and longer neck and better stance and a different gait. It's still a Doberman Pinscher. That's microevolution. No one's denying that exists. We all know and acknowledge that. But what we never see is a dog convert into a chimp somehow. But Darwin told us that essentially we would find these in the fossil record. And he said once we dig down deep enough, we are going to find a plethora of what he said on The Origin of Species in the book that he wrote, of these transitional species, because the dog's not just going to jump to the chimp. First, it's going to have to develop all these unique, subtle characteristics, and over time, that's going to add up into a completely different animal, a monkey. But what we have never found in the fossil record is one of these transitional species. This was one of the major talking points in the class I took. They're like, we need help. We need to go out there and find these transitional species. So here's what the scientists have done. They have created transitional species from two different animals. They've taken fossils from a horse and from a dog, and they've combined them together and they put them on display in museums and say, here is an example of a transitional species. They've admitted that this is what they've done. They still have them on display. These are the lengths that they will go to lie about the theory. They are manufacturing evidence that they've even disclosed they manufactured, yet it's still on display as an example of a transitional species. They claim that they have 6 examples of transitional fossils, where one animal is now slightly different now you can't really call it a monkey. Now it's something a little bit different because the anatomy of the bones are a little bit different. How many if any of those are real, we don't know, because they've lied to us all along the way by taking fossils from different animals and combining them together and acting like it's one animal find that they found. But what Darwin said is we should find a gajillion of these in the fossil records, because it would take so many subtle changes to get to the next species where you could say, well, this is now a completely different species. We don't see any of that. None of it. So until they magically find a gajillion fossil transitional species, they could throw evolutionary biology out the window. That idea is macrobiology. One species jumping to another. It's not a dog anymore, it's a horse. It's not a horse anymore, it's something else, an elephant. No, we don't see that. Macroevolution, we don't see examples of that. So when Tyler would tell his biology teachers at school, well, the difference between macrobiology and microbiology, hey, we all admit microbiology, microevolution exists. Macroevolution, there's no examples of that. And it blows the teacher's mind. They're like, The reason why I know is that Tyler's baseball teacher taught physics at high school, and he would be in the break room, and the biology teacher's in there talking to all the other teachers going, got this kid in my class, and he's telling me about macroevolution versus microevolution. I don't even know what he's talking about. And all the kids are listening to him and talking to him. He's like the teacher out in the hallway. That's exactly what went down. And then at baseball practice, the baseball coach is telling us, and we were all laughing and had a good time about it. He's like, it was hilarious. He's like, you blew her mind. She had no idea. So these, and this it doesn't take a PhD to understand this stuff, but this is really important stuff. We're way over time. I apologize. Let's end with a closing prayer.